Hi everyone, my name is Bill Wysocki. I am the lead for the user services team at the Genomic Data Commons. I'm here with my colleague Tara Lichtenberg, who is the clinical data curator of the Genomic Data Commons. And today we'd like to talk to you about the GDC Biospecimen Clinical and Experiment Data Model. Here's a quick outline of the webinar agenda that we will be talking about today. First, we're going to go over the GDC data model and have a quick overview of the submitted data portion of the data model, which contains the data that is submitted by external users, and the harmonized data model part, which contains the data that is harmonized and generated by the Genomic Data Commons. The next part is going to be on the curation and the guiding of the clinical data submission, which will go into detail about how our data model changes fit into that. And then we'll have two quick demos. One on the overview of the GDC dictionary tools. So there are two different tools, the dictionary viewer and the search tool, which will be demonstrated. And then a quick demonstration on how to find clinical and biospecimen data and different strategies for doing that. We will be taking questions at the end, so as you have them, please type them into the chat field, and they will be addressed at the end of the presentation. So the GDC data model is designed to maintain metadata integrity, so making sure the data is submitted in the correct place. Availability, making sure users know where to find the GDC data, and consistency, making sure that the GDC data is the same if data is the same. This data model accommodates biospecimen data, which is represented by the gray circles, clinical data, which is represented by the purple circles, and metadata for molecular files, which is represented by the red circles. Biospecimen refers to data that is collected from biological specimens that came from a particular patient. Clinical data refers to data that came directly from a patient in a clinical setting, and molecular data is the data that was produced during DNA, RNA, or other types of sequencing. The GDC uses a graph model, which is made up of two different aspects. There are nodes which contain information about each entity. These can usually be represented by something like a JSON formatted file. And then there are edges, which connects the nodes together. As you can see by the way this data model on the right is outlined, different node types can be only associated with other types of nodes. So only certain node types are only allowed to be connected to certain node types. So for the next slide, I'm going to show you the basic data model for the submission portion of the entire data model. So this is the part that a submitter would upload to submit everything from a case all the way down to a FASTQ file. Each of these entities are dependent on the one above, and we sometimes refer to that as a parent-child relationship. So in this case, the case is the parent node to the child sample node. And the same is the case for the parent sample node being parent to the child aliquot node. I'd also like to add that this is a very zoomed in and abridged version of the submission data model. There are lots of other clinical nodes. There's many other biospecimen nodes that are not included here. So the first portion of the submission data model I would like to tell you about is the case. The case usually represents a single participant in a study, and I say usually because it can sometimes refer to something like a cell line and not a living patient. The case itself actually contains a minimal amount of clinical data, which includes the primary site, which is the anatomical site where the primary tumor was identified, and the disease type, which is a high-level histological classification of the primary tumor. The case is also the anchor for the rest of the clinical data. So lots of clinical data can't be attached directly to the case because of many-to-one relationships. For example, if a patient went through 5 different treatments, they would need 5 different treatment values, which couldn't fit on one node in the graph database. It would need to be associated with many nodes that are attached to one case. So like I was saying just a minute ago, the case is where most of the clinical data is anchored. And this one in particular is the demographic, which, as you may suspect, contains demographic information about a particular case. Within the demographic entity, there are some important fields which include vital status, whether the patient is dead or alive, the gender, or more realistically, the biological sex of the patient, and the patient's race and ethnicity. Now going into a biospecimen type entity, here's the sample, which is a child to the parent case node. This represents tissue that was removed from an individual. This can be a blood draw, a cheek swab, a tumor excision. These are all valid examples of what a sample would represent in the GDC. Two very important fields that are required on the sample entity are the sample type, which is a representation of what the sample is. So this would tell you if it's a cheek swab or a blood draw. It would tell you if it's from a tumor or from some other some other anatomical part. The other important field here is the tissue type. So sometimes the sample type can tell us whether it's a tumor or normal, but sometimes it can't. Like if it was just a blood draw, we wouldn't know whether it's tumor or normal. So filling out the tissue type field is very important for submitters because this tells the bioinformatics pipelines whether it's a tumor sample or a normal sample, which is important when variant calling is done. So the pipelines know which alignments to pair with which alignments. Now, the aliquot here, this represents a molecular extraction from a sample. This can be a DNA extraction, an RNA extraction. In the future, we plan on having more protein. And this is used as a unit of harmonization. So all read groups associated with one aliquot within one experimental strategy are processed together. So if I'm going to go into read groups in a second, but the idea here is that if you had 5 whole exome read groups attached to this aliquot, those would all be processed and aligned together. And if they were spread over 5 different aliquots, they would be all processed separately. Now onto the read group. This usually represents one lane of next generation sequencing data. It can also represent part of one lane. So for example, in the case of multiplexing, one of the multiplex barcodes would be one read group, but it never would refer to more than one lane of Illumina sequencing. Read groups in the GDC are actually processed separately to take batch effects into account. This is why the GDC doesn't support, concatenating a bunch of FASTQ files from a bunch of different sequencing runs all together because of batch effects. And the GDC aligns the read groups all separately and then merges the alignments. Some important fields that the read group needs are the library strategy, which tells the bioinformatics pipelines how it should be processed. Is it a whole genome? Is it a whole exome? Is it RNA-seq? So the bioinformatics team will know which pipelines to actually run based on this strategy. The library selection field represents how a full extract is whittled down to focus in on some subset of the genomic aspect. So, for example, poly-A selection, you only want to look at the protein coding genes. Or RNA depletion, you want an RNA-seq without the ribosomal stuff there. Hybrid selection, using a target capture kit, which brings me to the third important field here, the target capture kit. This is important in whole exome and targeted sequencing because this tells the bioinformaticians which regions of the genome to focus variant calling on. In the case of something like whole genome or RNA-seq, this actually wouldn't be important, but we require that it is filled out as not applicable, just so that it never gets forgotten in the case that it is needed. Lastly, on this basic data model for the submission portion is the submitted unaligned read. This one's different from the others in that it represents one FASTQ file associated with one read group. Now, I bolded the word file there because this is the first entity that we've been looking at that actually represents a file. Some important fields here include the file name, MD5 sum, and file size, which are provided in advance of the file being uploaded so that the GDC can validate against the expected versus the observed values for these fields. So if you upload a file size of 10 bytes and then try to upload an actual file of 10 gigabytes, this will throw an error. The experimental strategy, like the library strategy before, actually lets the bioinformatics pipelines know which software to run. So in addition to all this metadata, this entity actually refers to a real FASTQ file that is submitted to the GDC to be processed. Now that's the end of the submission portion of the GDC data. However, I want to let everyone know that this is by far not the entire thing. There are more submittable data types. There's additional biospecimen elements. For example, between sample and aliquot, there are sometimes portions or analytes that can be submitted, and there are many more clinical types, such as diagnoses, treatments, molecular tests, etc. The next part of the data model I want to talk to you about is the harmonized part. This is the part of the data model that houses the generated data by the bioinformatics pipelines. So the thing about this part of the data model is that it represents logical harmonization steps. So these are connections between files and workflows. So you have a file which is processed using a workflow, which results in another file which is processed using another workflow, and so on. So it doesn't have an exact one-to-one match between the actual bioinformatics pipelines, but it does have enough representation to give the user enough information about how these files were processed. So let's build on that the same way that we did um, the submitted data model. This is the harmonized data model. Um, so we're going to skip the submitted unaligned reads because we already talked about that. But these are the submitted FASTQ files, sometimes BAM, but usually FASTQ. And the alignment workflow represents the workflow that aligns these type of files to the reference genome, which is GRCH38 in all alignments in the GDC. The two most important pieces of information in the alignment workflow are the software that's run, so which alignment software was used, and which version of the software was used. We want to be specific about which exact version was run for every particular file. Next are the aligned reads. This represents the aligned read groups to the reference genome. So all the FASTQs are aligned to a reference genome, and this is the file. These are the alignments that are downloadable on the GDC data portal. So when you go to the GDC data portal and you search for data type aligned reads in data format BAM, these are what you're looking at. Now, aligned reads can be done for many different experimental strategies, but in this case, I'm going to go down the whole exome path. So the somatic mutation calling workflow represents the process in which one variant caller assesses variants from a tumor normal pair. Note that if all 5 variant callers are used, then there will be 5 somatic mutation calling workflows attached to an aligned reads pair. This type of entity also contains the software type used, which is very important in this case because there's multiple variant callers, and the version of that software. Now, attached to the somatic mutation calling workflow is the simple somatic mutation. This represents a set of raw variants that was called by one somatic variant caller. So if you have 5 different workflows, there will be 5 simple somatic mutations. These are sometimes referred to as raw VCFs, and this can be found in the GDC data portal under the filters data type, raw simple somatic mutation, and data format is VCF. So that's the end of the part where I introduce the 2 parts of the data model. I also want to bring to everyone's attention that this one actually, in addition to having different paths, so for example, aligned reads can be attached to an RNA expression workflow if it's an RNA-seq alignment. This particular pathway actually goes a lot further, in which a simple somatic mutation is annotated and eventually converted to a MAF file, all of which are available in the GDC data portal. I decided not to include those because they would be very repetitive. So one question that we at the GDC get a lot is, how do we decide which data model changes are made? So how do we make this consideration? And I'm going to go over a very basic format, but Tara goes into a lot more depth about this. So in a basic sense, the GDC data model changes are made to accommodate new submission projects. So new projects coming in from new submitters and new harmonized data types. So if the GDC knows that a new data type is coming down the line, the GDC data model will be changed to accommodate that, so it will be available for submitters. When a change is requested, the following questions are considered. So one, is there currently a feature in the data model with the same function? We want to reduce redundancy. Like, for example, if a submitter came to us and said, "Hey, we have a lot of drug information. You should make a drug entity," we would tell them, "Well, there is a treatment entity, and on that is a therapeutic agent field where most drugs can be reported." So we wouldn't want to have the same data living in two places. The other question, the other two that kind of go together is how important and numerous are the data and how many projects will use the data. So if a project had a very important test results, but it only pertained to two different patients, the GDC probably wouldn't make a data model change for that instance. And if no other projects used that type of test, that would make the case further for not making the data model change. However, when a data model change is rejected or just not made, the alternative here is that the biospecimen and clinical supplements are available for submission. These are files that can be in TSV, JSON, XML format that submitters can upload to the GDC, and these will be made directly available to the public. So any type of clinical attributes, any type of biospecimen attribute, it doesn't matter if it's in the GDC data model or not, they can be put into these clinical supplements so they won't be withheld from external users. That's all I have to say about that. I will turn it over to Tara for some more talk on clinical entities.
Thanks, Phil. Sharon, can I get the ability to share? Okay.
Yes, Tara. Let me make you.
Got it. I think Bill just had to stop. Can you see my screen?
Yes.
Okay, great. I'm going to walk through like Bill said, the clinical side. Before I get started, I wanted to point out that I didn't include the word harmonization in my presentation at all, but it's very applicable on the clinical side too. So Bill spoke to the harmonized sequencing data. The effort that we're making to curate the data on the clinical side is also with harmonization in mind. So we also don't want to be redundant on the clinical side. So I'll start just by working through this real quick workflow, and then I'll go into more detail about each of these steps. So the first step when submitters are providing us with clinical data, they either need to get us what I'll call source data, and that can be in the form of a data dictionary or data collection forms or example data, subset of their existing data. And that process starts with us at the GDC, typically myself, creating a mapping file for them, proposing mappings, and then working with them. It's an iterative process to get to a final mapping that all parties can agree on. A faster path typically is when the submitter, they go out and access the GDC's data dictionary and they create a proposed mapping to start with. And that typically speeds things up, but sometimes data sets are so large and laborious, it's too much effort at first pass for the site who is less familiar with the GDC dictionary, obviously, than we are to make that first pass, but that typically is a faster path. Once the changes are approved by all parties, and I'll walk through each of these steps in more detail in a minute, the updates needed for the dictionary are determined, those updates are made, the final submission files can be prepared, and then files are submitted to the GDC. So like I said, data can be provided in many different formats up front. The more information we have about a data set, the better. But like I said, limited data sets, so smaller data sets, like Bill said, the required information is the histologic diagnosis of the patient, the site of disease, and then some limited demographics information, age, gender, race. That's what we really want for all cases. And groups who have a small subset of clinical data that mapping is relatively easy and can take less than a week to complete. But other groups have really large sets of clinical data, and they need more guidance on how they can make sure their data fit with the GDC. We determine what needs to be added to accommodate their data set. And those mappings, figuring out those mappings can take weeks and sometimes months, depending on the availability of all parties involved. And then I wanted to point out that if dictionary updates are needed, it could add significant time to the process. And I'll talk about our process for dictionary updates at the end here. So if a submitter was to propose their own mappings, they would start by going to the GDC dictionary. And I have a picture here of the dictionary viewer. They can select on the top right whether they want to use a TSV or a JSON format. And for each of the categories, you can see the clinical category here includes the nodes that Bill described. They're listed here, demographic, diagnosis, etc. If you were to click on the download button that I have circled here, a zip file would be downloaded with a TSV or a JSON template for each of these nodes. And so that can be used to create the proposed submission. And so this is a file that we received from Beat AML, one of our studies for leukemia, and they provided us with this is one of several files that they provided us. This one we didn't have any issue with. They did it on their own, and it was ready for submission right away. They submitted several files, and we had some back and forth on others. But once this was submitted, any user can now go to their project and download the JSON version of this on the GDC's portal. And that's what I have here in front is the JSON version of this file. And that's on a single case, obviously. The benefit of a TSV is that you get multiple cases per file. The JSON is per case or per node. So if the site does not do their own mapping, they provide us with source data. And this is an example where the site did a hybrid of doing their own mappings and asking for some guidance. And so we had access to, this is for a Burkitt lymphoma study, BLGSP. It's an NCI study, and we worked with the group who was submitting data. They provided us with a spreadsheet with all of the questions from their case report forms, which I have a picture of in the background. As you can see, it's nice to have as much data, as much information about the data that were collected as possible, so that we get a better understanding of how the source data can be mapped to the GDC dictionary, or how we add new properties and accurately represent the data that were initially collected. So we worked with them based on their initial suggestions. We gave them a lot of feedback using this Excel file, and then eventually they created TSV files for upload to the GDC. I wanted to give some specific examples of how we work through these sort of slight differences in data sets that come in. As anyone who's worked with clinical data knows, most data sets have synonymous terms, but they're often represented differently. So I just wanted to point out a few specific scenarios where that's happened. The first one is an obvious synonym. So the sites wanted a new property for venous invasion present. We already had vascular invasion present, and so we all agreed that these are synonymous and that the venous invasion could be mapped to vascular invasion. The next example are the submitter's data set included several yes, no, or Boolean style questions for the outcomes of pregnancies, and they wanted new properties for this. We decided that we wanted to, so I don't think I mentioned this specifically yet, maybe it's the next slide. When we're adding new properties, we want to consider both the current data set that we have already, so existing data that have been submitted, as well as what's being requested, but also future submissions. So what is this going to look like in the future? If we were to build out each one of these pregnancy outcomes as their own question, and then we had data that related to pregnancy outcomes come in the future, it would be much more difficult to map data to each, say, the date of a pregnancy or some sort of temporal information that came in about pregnancies. We would want to make sure that we accounted, that we were able to bring those data in together in a way that was sustainable in the future. So we created this property pregnancy outcome that has each of these variables, live birth, miscarriage, et cetera, as enumerations. And then in the future, if property is suggested about a pregnancy that can be related to the outcome, we can create multiple nodes to capture each pregnancy and any kind of data that has a relationship to that property. So that was the second one. And then the last one I wanted to point out was we created a new node. This was the last new node we created on the clinical side for BRAF expression by IHC. It wasn't necessarily this question exactly, but we were getting in a lot of molecular test information was generated on the clinical side or was generated prior to the sequencing that was done for the study that was submitting data. And so we wanted to make sure that we could bring these data in. We wanted to make sure we could bring them in a way that could potentially be compared or at least referenced against the data that are harmonized at the GDC. So those variant calls that are harmonized by the GDC's pipelines. And so we created a new molecular test node that can capture clinical molecular tests. And here you can see that gene symbol, molecular analysis method, and test result can pretty much bring in everything for this question. So like I said, when we're deciding what elements need to be added, someone during our practice run of this demo pointed out that it is both an art and a science. We need to make sure that we are accommodating the submitter, but we also want to make sure that there are standards. There are a lot of sort of aspects going in and thoughts that are put into adding a new property or properties. One thing that we need to do is search our own library, or our own dictionary rather, and as it grows, it becomes more difficult to remember or memorize or know what variables we have, what properties we have in our dictionary. And so the Center for Bioinformatics and Information Technologies Enterprise Vocabulary Services, it's a mouthful, helped us build a tool that searches the GDC's dictionary. And so first step is to make sure to search our dictionary and make sure that there isn't anything already that already exists. I advise folks who are interested in GDC data to use this search tool. It's really great. Bill will give a quick overview of the tool, but in a future webinar, he may go into more detail about this, so keep that in mind. Also, the NCI Thesaurus terms browser is a really great resource as well. We are currently adding NCIT terms to every enumeration in the GDC, so that is an upcoming goal. We started that process in our last release, excuse me. And the NCI Thesaurus has a really vast, a really broad and vast group of terms that can be used for studies that aren't just cancer related. They aren't just oncologic in nature. It's a nice browser. I also recommend you check it out. So we make sure that if we have a request and it already exists in NCIT, we want to use their terminology so that we're not recreating the wheel. And on that same note, if there are other resources, for example, our site of disease and histology terminologies are based on the International Classification for Disease, their oncology arm, the ICD-03 terms. We want to make sure that we use resources like that have already been created. And so if there is a standard or a classification that we can point to and that we can call out in our description of the property, we will do that. And then just so everybody has an understanding that the dictionary updates are very much vetted and reviewed before they go up, I wanted to just walk through this initial process. So the green are things that we already talked about in the prior slides. So the initial mappings that are done both by the GDC curator, myself, and the groups who are submitting the data. Any update to the dictionary that comes out of that is reviewed by GDC leadership, including Bill and others. As long as it passes that, they go to a larger discussion group. We have a working group that reviews every dictionary release that's going out. And so if that gets reviewed and approved, then it goes to the team to update. So we update the dictionary in GitHub. So if you're interested in the dictionary, the minute details of the dictionary, you can access that at GitHub. If any of these approval processes are not passed, we just walk them back through and make sure that we make the appropriate changes so that folks are on the same page and approving whatever needs to be done to the dictionary. Our QA, once it's in GitHub, we review, and all of this is tracked. We use Jira tickets to track all of the updates. Our QA team reviews the updates. Once they make an approval, the dictionary is released to the public, and then submitters are ready to create their final submission forms and files and submit. I just wanted to point this out also because it's not changes to the dictionary don't happen quickly. So we make dictionary changes about every quarter. So every few months we'll make dictionary changes, and they can accommodate. We make dictionary updates both to accommodate submissions, but also we usually have initiatives like the one I described, adding NCIT terms to enumerations. There are things that we are doing constantly as well just to update and improve the dictionary itself. But if updates were made based on specific requests, we will communicate those directly to the submitters. If, and this does happen, if a change that was either requested by a submitter or something that we are doing to just improve the dictionary or the data model, if that affects a submission that was previously submitted, we will work with the submitters who initially submitted the data to provide a proposed update, and typically we are able to do that so that they don't have to take any action on that. But if they do, then we'll work closely with them so that the burden is low for them. And then information about the dictionary is available for anyone. So if you guys, I believe, Bill can correct me if I'm wrong, but I believe if you're signed up for updates from the GDC, you'll get an update when there is a dictionary release. There on the doc site, which Bill I think will point out in a minute, there is a place where all of these dictionary updates are laid out in great detail. And then once any updates that are needed are completed, as I said, the submitter can complete the final submission files and submit them using the GDC API or the data submission portal. And I'll send it back to Bill.
All right, thank you, Tara. Let me go ahead and share my screen. Okay, so now I'm going to go into a demo on some of the tools the GDC has to more easily view the GDC data dictionary. Now, one question you may have is, what's the difference between the GDC data model and the GDC data dictionary? And the answer is that the GDC data model is more of an abstract representation of how the database works in the GDC, and the data dictionary is kind of the real-world implementation of how data is submitted with the data model rules in place. So this is the GDC documentation website, docs.gdc.cancer.gov. And if one were to go to the data dictionary tab and click on Viewer, this will bring you to the data dictionary viewer. Some may be familiar with this, but this is a dynamically produced, a dynamically generated website that pulls from the current dictionary and puts it into a more readable format. So first, maybe we can go and look at one of these clinical elements, the demographic like we were talking before. This tells the submitter what they need to do to get a demographic entity into the GDC. So if you want to hear more about the submission process, there was a past webinar about it that you can check out on the webinars website, but this will go more into just the dictionary viewer itself. So some of the most important things here are, one, it tells you what the entity needs to link to, and in this case, it's a case. If you don't have a case, you can't submit a demographic. Then the properties that can be listed, or sometimes people might call them fields. I do a lot. Properties or fields are listed. And as you can see, the required ones are listed first. So these are all the things that are required. Um, some of them, on some of the properties, something like unknown is allowed here, which simply means that it's okay if you don't have the information. We just want you to verify that you don't. So um, each of these properties has a list of enumerations, so a list of allowed values, and some are a little more flexible. Like for example, the agent index has to be an integer. Whether or not an age is obfuscated is a Boolean, true or false. And we are actually doing our best to remove um, the raw string fields because um, those can be hard to normalize. Um, but there are still some of those left. Next, I'm going to show you an example of a biospecimen uh, entity. As you can see, the sample can be attached to a lot more things. Um, a sample can be attached to a case, a diagnosis, another sample. This is usually in the case of a cell line being derived from somebody's sample, and a tissue source site. So the two fields that I was talking about earlier in the presentation, the sample type and the tissue type, are outlined here. So the tissue type has some very specific values because this just tells the bioinformatic pipeline, is it tumor, normal, and these are for some special cases. Sample type gives you a lot more detail about what the sample actually is. So it can be a primary tumor, it can be a metastatic tumor, that kind of information. Lastly, a feature that I forgot to mention before is I want to go over the aligned reads. Now, the aligned reads entity is an actual file. This requires information for the portal. So for example, the data category and data format, those show up on the portal filters. The experimental strategy, in addition to telling us how it was processed, it does also show up on the portal filters. And then the file name, file size, and MD5 sum are used for validation. Now you'll see there's a bunch of fields down here below, and these are actually produced by an internal quality control setup. And while not all of the fields are populated currently for some of the older alignments, the newer alignments do have most of these values. So for example, the contamination, so the fraction of reads coming from cross-sample contamination are reported in many of the newer alignments, as is the total number of reads, which I know is very important for some users. So that's all I have to say about the data dictionary viewer. This is basically just a nice representation of the raw dictionary looks like. Oh, one more thing that I want to bring to your attention that I think Tara talked about a little bit before is the download template function. So whether or not you like TSV or JSON, you can select and then click download template, which will provide a template for submission of all these properties. The next thing I want to go into is the dictionary search tool, which on the doc site you can get to by going to Data Dictionary and then clicking on the search button. So this is a very advanced and nice tool that I'm only going to delve into the surface here because there's many features in this tool. This is used for somebody who's kind of a beginner in the GDC. In this case, let's say I had a patient with sarcoma, and I didn't know where to put that value. Do I put it on the case entity? Do I put it on the diagnosis entity? I'm not sure. So I'm going to type sarcoma into this. You'll have some nice autocompletes that happen, and click search. So there's three kind of results tabs here. There's the values, the properties, and the dictionary. So the values will look for the term sarcoma in the allowed values for a field. So the tumor code can be these particular values. The disease type, like I told you before, was a high-level histological value. It shows that sarcoma can be put into this one too. Additionally, the primary diagnosis would be the best place for it because I only have sarcoma. I don't know which specific type it is, so I would put it under sarcoma, NOS, or not otherwise specified. Now, let's say I had the smoking status for a patient and I knew how many cigarettes per day they smoked. So I could search for cigarettes in here, and I would search for the properties, and here we go. There's one specific property, is cigarettes per day, and this is what I would submit to. If I wanted something more general, I could search for tobacco, and more tobacco-based fields are revealed here. Also, the dictionary results are just kind of gives you the same information, but in a bunch of drop-down menus as the dictionary viewer. So I know that if I look for something tobacco related, it's going to be clinical, and it can be either on the exposure entity or on the follow-up entity. So on the follow-up entity, there's a risk factor property that has three different tobacco related field or values, and the exposure entity has many different types of fields and values that include the word tobacco in it. So, like before, I can go into a lot of detail about this data dictionary search tool. We don't have the time right now, so a future webinar might cover this. Lastly, I want to go over really quick some strategies for searching for clinical data and biospecimen data. So you log into the GDC and you want to download a subset of files for some sort of clinical aspect. So let's say you only cared about the disease type and you only wanted something from us, and you only wanted data from males, and you only wanted patients who were still alive. This is how you could filter down by that. But it's not always that simple. Sometimes fields aren't there. So let's say I want a particular tumor grade that I'm searching for. So I look down on the case entity fields, and I don't see anything about tumor grade. The first place to look is this add a case biospecimen filter. This will tell you exactly which fields are available to query by, but aren't actually in the default view. So I can type tumor grade, and here we are. And now we can filter for tumor grades. So I click high grade, and this appears as a filter. And this filter can be gotten rid of simply by clicking this X here. Lastly, if these indexed fields, these queryable fields, never shows you the value that you're looking for, the last place to look is in the biospecimen and clinical supplement files, which can be found under clinical supplement and biospecimen supplement, which are under the data type. If you look under the formats, most are the XMLs and the SSF XMLs. And if you were to download them by XML, they would be on a case-by-case basis. So here's all the biospecimen information for this case. Here's all the clinical information for this case. And it can be filtered down by project, but this can kind of sometimes be hard to download every single one for 1,000 cases and parse out all the XMLs. So an alternative we have that was actually generated from the XMLs is the BCR Bio tab. These were generated for the TCGA project, so these are the only place these will be found, and they are aggregated for all of the patients in a particular study. So in this case, if we wanted to look for only those in breast cancer, we would click on TCGA BRCA. This would give us a list of all of the biotabs for TCGA BRCA. So there's different types. There's analyte, which gives you all the information about the analytes. There's the drug. There's the slide. And the one we're going to look at now is the patient biotab, which I downloaded and put into a Google Sheet so I could show it to you. And as you can see, it's in a nice tab-based format. It's a TSV, so these properties can be pulled. They have some pretty project-specific fields in it. So if you wanted to look at something like the ER level, cell percentage category, or breast carcinoma immunohistochemistry ER positive finding scale, these type of things wouldn't be added to the GDC data model, but they would be available here. And as you can see, not all patients have them available, but those who do are reported here. Okay, that's all I have to say about querying clinical and biospecimen entities, and that is the end of this presentation.
Thanks, Bill. There are several questions. The first question is, where do portions and analytes fit into this? Are they an alternate route between aliquots and sample?
Okay, that's a good question. Real quick, can you see my PowerPoint? I'm sorry.
No, it still has a spreadsheet.
Okay. Here we go. Okay. So my understanding, and please step in, Tara, if you have any more details, is that you have sample, and if all the information you have is that DNA was extracted or RNA was extracted from that sample, you can connect an aliquot directly to that. The TCGA method for data model structure has sample with a portion attached to it, which represents a part of the sample, like as if a piece of the sample was cut off, that would be the portion. And then below the portion would be the analyte, which represents the full extraction of the analyte, of the DNA or the RNA. And then when that analyte is diluted, presumably to something that would be processable that can be processed with a Illumina prep library, that would be the aliquot. So the more standardized concentration of the analyte would be the aliquot.
That's the right path, Bill. Yeah, that's correct.
Okay, great. We have a comment that the presentation is fantastic for explaining the system. Is there a way it will be made available, or is there another resource will all of this information that would be recommended to be used for reference? And the answer to that is yes, this will be available, the video will be available on the GDC website under support, and we do have the data dictionary on the doc site, as well as the data dictionary search tool. So there's several areas on our documentation site as well that does explain this. Bill, did you have any further comments regarding that?
No, just take a look at the video when it's out and look through the documentation website. We keep that relatively up to date, and it's updated fairly often.
Okay. The next question: is it possible to download all normal tissue slides, the SVS files? As you said, it comes with a combination of sample type and tissue type. What is the best practice for field selection to get all normal slides? I have seen normal and frozen section.
That's a good question. And currently in the GDC, most of the slides are from TCGA. TCGA slides are more were imported before the tissue type field became a thing. So you would query for sample type and then select all of the normal sample types, which would give you those slides. So the same way that I queried before for tumor grade, you could query for sample type. And this would give you a list, and you can just select the normals.
Okay. There was a question of whether the CADSR was used to inform the data model and data dictionary.
Yeah, I left that out. That's a good. I should have mentioned that. So all of our the majority of clinical properties have a CADSR code, and within the CADSR, they've assigned NCIT terms as well. I just wanted to point out that their NCIT terms may be slightly different than the NCIT terms we are assigning because we are vetting those with a little more detail.
Next question: if parsing TCGA clinical files, properties associated with follow-up appear to be available, but they appear to not be available from querying the data model. I think I remember that there are no follow-up nodes for TCGA data at this time. Is this true?
So
That is true. Sorry,
yeah.
Go ahead, Tara.
Yeah, I'll just take this because I curated this. So that's true that there are nodes, but the days to last follow-up, anything that would have been on the follow-up node, we looked at all of the follow-up data that were submitted for TCGA, and we used the latest date. So if a patient had enrollment information for TCGA and then they provided follow-up information, in the GDC, we're using that follow-up information for the properties that are on the diagnosis node currently. So days to last follow-up is on the diagnosis node currently. Whether they received radiation or pharmaceutical treatment is on the diagnosis node. Those are the most recent data points that you'll find in the TCGA data, if that makes sense. It's still being discussed. Your question about whether there will be more available in the future, I think that may be another question the same person had. I think that's still on the table. It hasn't been totally taken off the table, but discussions are still being had around that topic.
Okay. The next question is, specifically, is the data model still in the process of being populated for older programs?
Oh, so that was the question I saw. That was, yeah.
Okay.
Yeah. I think that's still being discussed, right, Sharon? Whether that will happen or not.
Yeah, for TCGA, yes. Does GDC create pseudo IDs for analyte and portion if not submitted?
That one, Bill?
Yes. So that does happen. If they aren't submitted, they do get these pseudo IDs that are used to kind of take the place that they would have been had they been submitted. It doesn't affect anything except for the appearance, and these pseudo IDs aren't seeable by the external user. They're just in there to hold, um, to be a placeholder so that all of the data is more or less congruent.
Okay. In many studies in medical imaging, classify the data based on project name. Is this an accepted method? Do we have a common subdisease in two different projects?
I'm sorry, could you repeat that question?
Sure. In many studies in medical imaging, they classify the data based on the project name. Is this an accepted method? I mean, do we have a common subdisease in two different projects?
Sorry, Tara, do you know the answer to that? Because I'm not sure.
I think I understand the question, but the user can reach out to user services or to us in another way if this isn't clear. But I think you're asking if studies have multiple diseases within a single study, is it okay to class for imaging? I'm not sure whether you're asking specifically for imaging or not, but I think it's important to note that in the GDC, we have study name, but we also have, so it's called project, I guess, if you look on the portal. But it's all of these are also split up into disease type and primary site. And so you can classify them either using the disease type, primary site, or like Bill mentioned in his presentation, there's even more, or in his demo, there's even more specific site information and diagnosis information using the primary diagnosis and the there's a couple different site properties on the diagnosis node. But if that doesn't answer your question, maybe we can go into that in more detail offline.
Okay. There's a question regarding clinical data submission, specifically the aliquot file. Is there a way to annotate aliquots that have more than one parent sample? They're cryopulverized within the same case. The GDC portal has issues warnings for such aliquots.
Oh, yes. So that's a good question. So first, they will be attached to multiple samples, and this is represented in the GDC data model. However, this also can be actually annotated. We have an annotation function that attaches to every node type, so we can annotate anything. So if you do have a submission project and you would like to annotate something, contact the GDC support email, which you can see at the bottom of this slide, and you can find it at gdc.cancer.gov, and we can help you out with that.
Okay. Is there a way to download a snapshot of the data dictionary with associated case or file IDs similar to the BioTab file shown?
So what we do have in the GDC is a function on the portal. I could show this real quick. Is if you add some things to your cart, and then you go to your cart, you can download a sample sheet, the clinical data, the biospecimen data, or the metadata, and this will give you a TSV or a JSON based on whatever set of files you're looking at. So that's kind of the way that we do have a customized, more or less biotab function, but it does, like you said, have to adhere to the dictionary. It won't pull in any values that aren't variable.
And there's one more question about parsing a list of XMLs and/or files from the REST API is acceptable, but would like to avoid overlay taxing your server. So I guess it's more or less a statement.
Okay, yeah, like if you download all the XMLs ahead of time in just one giant chunk, it would only be, my guess would be in the tens, maybe 100 megabytes, and then you can do that locally. Um, and wouldn't need to hit our API so many times. So that would be my suggestion.
Okay, and that was the last question. I believe we're also out of time. Thanks everybody for attending, and please stay healthy. Have a great day.
Thank you, everyone.
Thank you.